Welcome to the deep dive. Today, we're wading into, well, one of the most clinically relevant and, frankly, rapidly changing areas of ophthalmology, age-related macular degeneration.
It really is.
And this isn't just a clinical challenge, it's a demographic one. I mean, we're looking at projections showing over 5 million people aged 60 and up affected by 2050.
It's a huge, complex burden. And especially when you look at neovascular AMD. Anti-VEGF treatment has been, absolutely revolutionary.
Oh, without a doubt.
But the complexity hasn't gone away.
Yeah.
We're still dealing with the immense pressure of frequent injections. And we know that those visual acuity gains, they tend to plateau or even start to decline after, 5 to 7 years?
That's the real clinical pressure cooker we're all in. That residual fluid, it persists in a staggering percentage of our patients. And the ultimate paradox is this. While we treat the leakage in wet AMD, nearly 40% of those same patients still develop geographic atrophy over 5 years.
It's the Achilles heel of the current paradigm, really. So our mission today is a high-density update on the pipeline that's designed to solve that very paradox. We're deep diving into a massive review that looks at everything up to 2024.
Everything. Gene therapy, cell therapy.
Complement inhibitors, novel combinations. We've done the heavy lifting so you don't have to strain your eyes keeping up with all the literature.
I see what you did there. So just to quickly orient everyone, dry AMD is the gradual one. It's marked by drusen, that slow progression to GA and irreversible cell loss. And wet AMD, or AMD, is the rapid, vision-threatening neovascularization, edema, and hemorrhage that's driven largely by VEGF.
And we really focused on sources that capture the full picture of what's being tried, which, and this is important, includes news releases right alongside the traditional peer-reviewed data.
Right, because that's where the cutting edge is.
Exactly. And that data you mentioned earlier really highlights why this innovation is so critical. Because treating the leakage with anti-VEGF doesn't stop the progression of the atrophy.
That's the paradox. So this whole pipeline we're about to get into, especially in the wet AMD space, is really a race to solve that long-term problem by increasing durability.
Precisely.
Okay, let's unpack this. The biggest headache in AMD for both the patient and for us is injection frequency. Gene therapy is aiming to provide some long-term citation relief.
Another one?
Yep.
Well, the goal is elegant, right? Use these highly efficient adeno-associated viral vectors, AAVs, to deliver a genetic code.
A single administration.
That achieves constant long-term anti-VEGF expression.
So let's start with RGX-314.
Okay, so this one delivers a genetic code for a ranibizumab-like antibody fragment. The key here is the delivery. It can be subretinal or suprachoroidal, and the results were, well, they were staggering.
How staggering.
They showed an 80% decrease in the annual injection rate. And get this, 50% of patients became completely injection-free.
50%. That fundamentally changed the entire patient care conversation.
It does. Then you have ADVM0202, which is designed to express the aflibercept protein. It uses a different vector, and an AV2.7M8.
Right.
And the durability reported in the OPTIC study was just as remarkable. We're talking an 80 to 98% reduction in yearly anti-VEGF injections over 2 years.
Wow. Any side effects that stood out?
The main thing was inflammation, some anterior chamber cells. But it responded well to topical steroids. It does raise that important clinical question, though.
How long do we need to continue prophylactic steroids after these therapies?
Exactly. But that durability is the game changer. And speaking of changing the game, this is where it gets really interesting on the gene therapy front. 4D-150.
This is the one that's going after everything.
It is. It is the first retinal gene therapy trying to inhibit 4 VEGF-related molecules. So VEGF, A, B, C, and also PIGF.
All from 2 transgenes.
All from 2 transgenes. That multi-target approach is revolutionary. It's trying to bypass all these escape mechanisms. And the phase 1/2 results showed that after a single IVT dose, about 80% of patients in the high-dose group were injection-free at 36 weeks.
So they're going straight for a non-inferiority study against standard aflibercept.
They're aiming high.
That 4-target approach, it really acknowledges that just hitting VEGF-A isn't enough for long-term control. So if gene therapy is solving the frequency problem with a single dose, what about stacking targets using combination proteins?
That brings us to IBI-other2.
Efdamar-FUSP alpha.
Yes. This is a novel bispecific fusion protein. It targets VEGF, obviously, but it also, and this is crucial, targets the complement pathway.
So you're hitting the angiogenesis and the inflammation at the same time.
Precisely. It works by binding C3b and C4b to directly reduce that complement amplification. The phase 2 data showed it was non-inferior to aflibercept, but the real clinical prize was the dosing interval.
Let me guess. Longer.
Much longer. Over 80% of subjects on the high doses could extend treatment to every 12 weeks. And the ongoing phase 3 STAR study is even looking at a 16-week regimen.
Q16. That is durable. And speaking of other targets, we should mention Restore It, a WNT pathway agonist. This one is going after subretinal fibrosis.
A devastating complication that anti-VEGF just can't fix.
Exactly. Restore It mimics Norrin, and it tries to restore the blood-retinal barrier. Early data showed improvements in retinal thickness and vision when it was used with aflibercept.
The whole focus is clearly shifting. It's moving from just suppressing leakage to, true long-term control and maybe even some tissue restoration.
So if viral vectors carry these inflammation risks we talked about, what's the next approach for that long-term delivery without them? I think we need to talk about TKIs.
Tyrosine kinase inhibitors, yeah. They're a great target for sustained delivery because they block multiple growth factor receptors, specifically VEGF and PDGF receptors.
Like AXPSLI or OTX-TKI.
Right. That one uses a bioresorbable hydrogel implant to deliver axitinib. The goal is sustained release for 6 months or more. Phase 3 is already underway.
And there's a similar concept with EYP-1901, which is a delivery system for vorolanib.
The phase 1 and 2 results for that one showed a significantly reduced need for extra anti-VEGF shots over 12 months. The whole segment really shows us the emphasis is on making the delivery itself less intrusive.
We can't overlook the advancements in the drugs we already use either.
Oh, absolutely not. Elay HD, the 8 milligram high dose of aflibercept, got approved last year. The Pulsar trial confirmed it can support dosing every 8 to 16 weeks.
Which is an immediate practical benefit for reducing clinic visits.
It is. And faricimab, targeting both VEGF-A and ANG2, has shown quicker retinal fluid drawing and fewer injections than standard aflibercept.
And then there's the ranibizumab port delivery system, Susvimo, constant drug distribution for 6 months or more via an implant. We do have to mention the voluntary recall it had in 2022.
A technical issue, yeah, with the septum. But trials are still ongoing to see how it performs long-term.
Okay, we spent a lot of time on wet AMD, but the real historic shift recently has been on the dry AMD front.
Absolutely. I mean, for our colleagues, the period since August 2023 is truly historic. We finally have approved therapies for geographic atrophy.
Finally. And they all rely on inhibiting the complement cascade.
That's right. The first 2 approvals are at last giving us tools to slow the progression of the atrophy itself. So we have pegcetacoplan, which targets C3. Studies showed it achieved an 18 to 22% reduction in GA growth over 2 years. And then avacincaptan pegol, which targets C5, showed about a 14% reduction over 1 year.
So I have a C3 and a C5 inhibitor, 2 different targets that are finally slowing GA. That's huge.
It is.
But what is the major clinical caveat we absolutely must be aware of when we start these treatments?
This is the critical safety note for every clinician. Treatment with both of these approved complement inhibitors carries an increased risk of the patient developing wet AMD.
So we're slowing the atrophy, but we might be increasing angiogenesis risk.
Exactly. Now, it's treatable with the anti-VEGFs we have, but it adds a layer of complexity to patient selection and monitoring. It's a trade-off.
Turning to the GA pipeline then, gene therapy is active here too, but going after a different target, JNJ-1887.
Yeah, this one targets the complement terminal pathway. It's an AAV2 therapy that enhances soluble CD59 to directly inhibit the membrane attack complex, the MA.
And the early results.
Phase 1 showed decreased GA progression in the high-dose group. A phase 2b study is ongoing now until late 2025, so it's the same theme of complement inhibition, but aiming for that single-dose durability.
And beyond gene therapy, cell therapy holds tremendous promise for GA because it's the only approach that aims to actually replace those lost RPE cells.
RG6501, which is an allogeneic RPE cell therapy, showed really compelling preliminary results in phase 1 and 2. Patients gained an average of 7.6 letters. And they saw structural improvements that lasted for up to 4 years. The potential for a single intervention to provide multi-year effects is, it's massive in a progressive disease like GA.
We're also seeing ASP7317, which uses human embryonic stem cell-derived RPE cells, recruiting in phase 1b. The ability to structurally restore the retina, even just partially, that moves us out of just slowing the disease and into potentially reversing some of the damage.
And finally, a couple of novel targets for intermediate dry AMD. There's rasuteganib, or Luminate, It's an integrin regulator for intermediate dry AMD.
Addressing oxidative stress. This one got quick results, didn't it? Phase 2 showed almost half the patients gained more than 8 letters.
Which is very impressive for a non-GA treatment. And then ALK001 offers perhaps the ultimate convenience, a modified vitamin A as a once-daily oral medication.
So it's aiming to slow the formation of those toxic dimers associated with retinal aging. That approach eliminates the injection burden entirely for early-stage patients.
It's just care right into the patient's home.
We've covered a remarkable breadth of innovation today. From single-dose gene therapies that promise almost permanent anti-VEGF expression to complement inhibitors that are finally measurably slowing GA. And all these novel drug delivery systems pushing dosing out to 16 weeks or even longer. The complexity now shifts back to us, the eye doctors, to decide which of these promising treatments is right for which patient and at what stage of their disease.
And that raises an important question the source article highlighted about long-term safety. It notes concerns in gene therapy, like pigmentary changes and inflammation. And we have to figure out, are these issues just intrinsic to putting these things in the eye?
Or are they due to insufficient prophylactic steroids?
Exactly. How long should prophylaxis be given? That's something we have to monitor very closely in the upcoming trials.
Absolutely. The source material we discussed today, dry and neovascular wet age-related macular degeneration, upcoming therapies, is linked directly in the description for you. If you found this deep dive valuable, please follow the podcast to catch our next analysis and maybe leave us a 5-star rating. We appreciate you joining us for this crucial update.
It's just a very exciting time to be practicing in this field.
Final provocative thought. Given what we're seeing with these combination therapies like IBI302 with its VEGF and complement targeting and the focus on broader targets like WNT agonists, could these new multi-target approaches also prove effective in treating other ocular diseases linked to inflammation, like diabetic retinopathy or uveitis? Could they extend their utility far beyond AMD? That's the question that I think will frame the next decade.